Hello, everyone. Thank you for being here today. We're very excited to have over 500 people from more than 23 countries join us from around the world. So to start today, and in keeping with the spirit of reconciliation, my slides to work, I'd like to take a moment just to acknowledge the traditional custodians throughout Australia, New Zealand, and all nations. We honour their profound connections to land and water and biodiversity and culture and pay our respects to their elders past, present and emerging. And we acknowledge the important role that Aboriginal and Torres Strait Island people continue to play within our community. So thank you again for joining us today. And for those who haven't met me, my name is Beryl Morris and I'm the Director of TERN, Australia's Ecosystem Observatory, where we're trying to help advance ecosystem science and contribute to the sustainable management of Australia's environment. Now, we are recording today's meeting, so I hope that's all right with everyone because the recording will be available to you later. Just a couple of words about TERN. We are a federation of the universities of Australia and CSIRO. And we also have connections across the world to a range of other groups. And we're funded by the Australian government through an NCRIS infrastructure grant with co-contributions from several of the state governments and lots of in-kind from people like yourselves. So thank you very much for making TERN possible. So today, I want you to meet the speakers of our talk, which is about the challenges of monitoring and measuring drought. So our first speaker is Georgie Foster, who's from Australian National University. Georgie is a climate scientist with a particular focus on understanding long-term variability in the Earth's water cycle. You'll be able to read more about Georgie in the bio that was sent out. Our second speaker is Anne Griebel from the University of Technology, Sydney, where Anne is a leading expert in ecological drought, which is very good for us, and long-term environmental monitoring. And our last speaker is Dr. Elisa Garola, who is part of TERN Australia, where she looks after the data aspects for our education and managing the long-term big data challenges that go with pulling together lots of data sets. So I'm not going to tell you very much more because our brilliant speakers are going to tell you all about things themselves. And so I'm just going to stop sharing and hand over to Georgie to take us away. Thank you.
Wonderful, thank you. All right, we can see my slides. Yes. Okay, so thank you very much for having me in this great lineup of speakers, and I'll be kicking things off talking to you about the very cheery topic of Australian mega droughts. So just to set the scene here, I'm showing a plot of rainfall anomalies over the Murray-Darling Basin, which I'm showing in this little map here on the right. On this plot, the green bars show years where there was more rain than normal over the Murray-Darling Basin. Brown bars show when there was less rain than usual. And here I've also marked major historical droughts in the Murray-Darling Basin, including the Millennium Drought here, which was the backdrop to my personal childhood. Now, the broad sort of current scientific understanding is that across most of southern Australia, droughts and their impacts are likely going to get worse with future climate change. But my work actually looks in the other direction, to the past, to understanding the full natural range of variability in Australian droughts. And this is because, you know, if we want to know how bad droughts could be in the future, then both of these things are equally important: climate change impacts and natural variability. But I'll start by taking a step back to ask maybe a more basic question, which is, what exactly is a drought? Well, this is the definition from the IPCC, which states that a drought is a period of abnormally dry weather long enough to cause a serious hydrologic imbalance. But then goes on to differentiate between agricultural drought, hydrological drought, meteorological drought, all of which are defined in quite different ways. So I'll actually be talking about maybe the simplest drought type, which is meteorological drought, which is essentially just periods of time where it rains less than normal. And one of the ways we can quantify less than normal is with this definition here, where we say that a drought starts with 2 years of below average rainfall and ends with 2 years of above average rainfall. And so I've marked in grey here all the years that would be classified as drought under this particular definition. And this is the definition I use for the rest of this talk. And before we move on, I think it's worth stating that, as you can see here, droughts really are a natural feature of the Australian landscape. In fact, many First Nations languages don't even have a word for drought as the climate hazard we consider it today. Many Australian plants and animals are very drought tolerant. It wasn't really counted as an extreme event until the past 200 years with Western style agriculture and built environments that are much less well adapted to periods of lower rainfall. Now, just before we really get into it, I've got another couple of definitions for you around how we classify climate variability. The first is internal variability, which is just the sort of day-to-day, year-to-year fluctuations in the climate system. It's why the weather isn't the same every day, every year. It has to do with things like ENSO. You can think like butterfly effect. And then we have external climate forcings. And these are things that change the overall amount of energy which is available to the climate system. There aren't a lot. Some are natural, like volcanic eruptions and changes in the amount of energy from the sun. But then there are also anthropogenic or human-caused external forcings. So things like greenhouse gas emissions, which trap energy on Earth, and aerosol emissions, which shield Earth from incoming solar radiation. Okay, so with that in mind, we want to answer the following question. Are the rainfall droughts we've experienced in the historical period unusual in any way compared with natural variability, or is there some influence of human-caused climate change? And also, does this small selection of droughts that I'm showing here in the historical record reflect the full possible range of what we could experience even without climate change? And it turns out this is actually a surprisingly difficult question to answer for a few reasons. First, that our observational record of rainfall in Australia is short. So at most 120 years or so. And as it turns out, this is not long enough to characterize the full possible range of internal variability in Australian droughts. Another difficulty is that all of our observations of Australian climate were taken during a time when human activities were already affecting the climate. So the Industrial Revolution was around the year 1850 or so, and most of our rainfall records in Australia are from about 1900 or later. Now, if we were in Europe or the US at this point, we would turn to natural archives of information about prehistoric climate. So that's things like tree rings, for example, where they can preserve information about climate variability as the tree was growing, which we can then use to reconstruct rainfall changes over past centuries. Unfortunately, in Australia, this type of data is extremely sparse. So we turn to the last available source of information about long-term climate variability, which is climate models. Just briefly, climate models are essentially extremely large sets of equations, which together describe the entire global climate system and how all the bits interact with each other. So they simulate both internal variability and also the climate system response to external forcings. So for example, climate models are used to predict or project how the climate system will change with future greenhouse gas emissions, with global warming. Now, there are a lot of climate models out there. This little plot I'm showing here shows a suite of 12 climate models where someone has run that model not forward, but back in time. So to simulate climate variability over the entire last millennium. More specifically, all the way from the year 850, which for reference was around the time when Vikings were plundering various parts of Europe. Also, algebra was being invented by a Persian mathematician. Another little bit of trivia here I've got written on the slide. The models are often, although not always, named for the research centre that developed them. So for example, here, the Hadley Centre in the UK, the Beijing Climate Centre in China, CSIRO here in Australia. And the point of all this is that we can look at drought characteristics in these models in the pre-industrial last millennium, so where we have 1,000 years of simulated natural climate variability, and then compare that with how droughts have looked since the Industrial Revolution. So since humans have started making really large-scale changes to the climate system. And that sort of answers two questions at once. First, how have Australian droughts changed because of climate change? And second, if we had 1,000 years of rainfall data instead of only 100, what other sorts of things we might see? And so when we're asking this question, what could droughts look like? There are quite a lot of drought characteristics that we can examine. I've listed them all here for you. Things like the total amount of time overall spent in drought, the frequency that droughts come around at, and the actual intensity of the drought, so how much rain is missing. But in the interest of time this afternoon, we'll just look at a couple of these, which is the average drought length and also the maximum possible drought length. In both cases, comparing those values in our suite of climate models in the pre-industrial last millennium, so the 1,000 years from the year 850 to the year 1850, with the values in the 20th century, so from the year 1900 to 2000. Okay. So first, we're going to dive in and have a look at the average length of droughts. So in this map here, I'm showing the average result from all 12 of those climate models that I showed. And where a grid cell is red, that means that in that place, droughts were on average longer in the 20th century than the pre-industrial. And in places that are blue, droughts were on average shorter in the 20th century. And so you can see that the darker red colours, which indicate a bigger difference, are really concentrated in southwestern and eastern Australia. So to just summarise this result, because of climate change, these models suggest that droughts are getting, on average, longer in the parts of the country that we sort of already think of as drought-prone. And we can say that this is because of climate change, because in these models, the only thing that really changes between the pre-industrial and the 20th century is the addition of those anthropogenic external forcings, like greenhouse gas emissions. Now, moving on from average drought length to the maximum possible drought length, on this slide, I'm showing in each of those 12 models and also observations in the top left across the entire country, the length of the single longest drought in that model's simulation of the 20th century. Another way of saying that is that these are 12 different estimates of how the 20th century could have looked within the range of internal variability. And so the longest possible drought here in these 100 years ranges from about 6 years up to sort of more than 20 years in some parts of the country. Now here I'm showing the same thing for the 1,000-year-long pre-industrial past millennium simulations. And you can see that across the board, the maximum possible drought length is much longer, sort of 20 to well over 30 years across the country. And this leads us to an important question. Why? Why were droughts so much longer in the pre-industrial than the 20th century? And the answer, of course, is that they're not. Well, not really. It's just that these extremely long droughts don't happen very often, much less than 1 per 100 years. So it is just by chance that we haven't experienced one of these mega droughts since the European invasion of Australia and the introduction of Western style record keeping. And so on that same theme, but now looking specifically at the Murray-Darling Basin in Eastern Australia, which is the main agricultural area of the country. So the bars here show the lengths marked at the top of the most famous or well-recorded historical droughts in the Murray-Darling Basin, from the recent short but very severe tinderbox drought to the longest recorded historical drought, the Millennium Drought. And now I'm showing the modelled maximum possible length of a drought in the Murray-Darling Basin if we had a full 1,000 years of rainfall records, not just 120. So this fat purple bar shows the model ensemble average estimate, which is about 26 years. The absolute top estimate from the models is the top of that thinner grey bar, way out at more than 35 years. The absolute minimum estimate is around 9 years. The keen-eyed amongst you will note that this is actually shorter than some of the historical events. That's in part just because of internal variability, but also because of that particular definition that I've used to identify droughts. But the real point here is that if we take that model ensemble mean, which is generally a pretty good bet, then we can say that the Murray-Darling Basin is naturally susceptible to mega droughts of more than 20 years, even before we factor in the effects of human-caused climate change. And this might seem extreme because such a thing hasn't happened in living memory, but keep in mind that the southwestern US, which has quite a similar climate to southeastern Australia, is currently in the grips of a mega drought, which has been going since at least the year 2000, so making that 24 years now. And as to when exactly we should expect a 20-year mega drought in the Murray-Darling Basin, I'm showing that here. So on this maybe slightly complicated plot, on the y-axis is droughts of a certain length, and on the x-axis is how often we might expect a drought of that length, like once every 100 years, once every 200 years, and so forth. And the different lines are the results from all the different models. So looking at a 20-year drought in the Murray-Darling Basin, depending on the model, we might expect one of these to roll around just naturally between once every 100 years to once every 800 years. For a 30-year drought, that would be anywhere from once every 450 years to less than 1 per 1,000 years. But my final point here is that even though these multi-decadal droughts are a natural feature of the Australian climate systems, and Australian ecosystems are likely adapted, well adapted to deal with very long dry periods, if a mega drought occurred today, it would be against the backdrop of global warming, which, amongst other things, can make droughts worse because the soil dries out more quickly, as well as making the impacts on people and the environment more severe because it's hotter. And of course, many of our natural environments already have other human-caused stresses to deal with, so it's possible that their resilience is decreased. So that's it from me. I'll leave you with these take-home points that I won't read out, but feel free to screenshot them if you like. I'm really sorry, I actually have to run off before question time. I'm actually attending a workshop this week. But if you have any questions, then please feel free to pass them on to the conveners in question time, or email me. I'll try to answer a few questions in the chat as well. So thank you very much. I'm going to stop sharing my screen and now hand on to our next wonderful speaker, Anne. Thanks.
Thank you, Georgie. That was a wonderful presentation. This one. Alrighty, I hope you can all see my full screen now. All right, if I don't hear complaints, I'm just going to start. Thank you as well for inviting me to the TERN seminar. My name is Dr. Engriebel, and I'm going to give a presentation on how we monitor ecological drought from plant to ecosystem scales. But before I begin, I actually want to acknowledge the Darug and the Gadigal people upon whose ancestral lands this research was conducted and where I'm giving this seminar from. I'd also like to pay respect to the elders, both past and present, acknowledging them as the traditional custodians of knowledge for these places. So let's start with highlighting some of the challenges that we face when we want to monitor drought in Australia's temperate forests. Well, first off, these ecosystems are actually characterized by really diverse plant communities that grow in very complex vertical arrangements, right? So that means we have a large mixture of species that is distributed across multiple strata in the canopy. And this actually might result in drought effects not being felt uniformly across species or across ecosystems, and that we cannot necessarily detect drought impacts without holistic assessments of ecosystem health in temperate forests. It also poses challenges when we infer dynamics from leaf to ecosystem scales, right? Or when we want to transfer dynamics from one temperate forest to another. So as Georgie already pointed out, the weather droughts are frequent and the weather is getting more extreme in recent years. So we have noticed very large seasonal and interannual variability of rainfall due to continental scale weather patterns lately. But as the frequency and the intensity of drought and extreme events in generally increases, even though they might not last long, they might still have prolonged consequences for ecosystem health and for long-term stability. So how resilient are temperate forests to drought? Well, that actually depends on a range of factors. And one of them is how well forests or plants in a forest can access below ground water. So rooting depth and the spatial arrangement of roots determines the access to soil water in the vadose zone, but the depth to the groundwater table also can vary substantially during a drought, and that can remove some stable water sources as the drought progresses. Further, community structure and composition can affect how well species within an ecosystem can cope with drought through competition being a factor. So with competition, I actually mean access to resources like light and water. And I also mean that it affects the microclimate that the different species experience within a complex forest strategy. Another confounding factor are plant water use strategies. So there are some species that prioritize carbon sequestration and they're more lavish in their water use, whereas others, they maximize the integrity of their hydraulic systems and they generally tend to be more restrictive in their water use. Another important factor is how healthy the ecosystem is when the drought commences. And that obviously depends on when was the last drought before then, how much time did the ecosystem have to recover, but also on the duration and the intention and the frequency of the drought that is affecting the ecosystem. So I'm going to present some of our observations here from the Cumberland Plain woodland, where we had multiple studies underway during the 2017-2020 drought, which Georgie nicknamed the Tinderbox drought. And that one allowed us to decipher how drought in combination with heat waves and with a biotic agent affected carbon, water, and energy cycling. Sorry, flip too quickly. So what I'm looking here is at the study in our Cumberland Plain woodland. And the Cumberland Plain woodland, for those that don't know, they are surrounding the Sydney Basin and they're very prevalent here. They are dry sclerophyll forests that are well adapted to seasonal droughts, to nutrient limitations, and to decadal fires. So they typically experience between 700 and 900 millimeters of rainfall, and Cumberland Plain woodlands host about 1,200 plant species. So that might sound a lot, but they're actually recently been classified as critically endangered ecological community, and that is because the urban development and the mixture of urban sprawl and land use change for agriculture have resulted in a severe fragmentation of the ecosystem. And now there's only about 13% of Cumberland Plain woodland remaining in the area here. So we've conducted the study at the TERN study site, which is a long-term monitoring site in a 190-hectare large remnant dry sclerophyll forest near Western Sydney University. And the woodland section that we've conducted the study in has been dominated by 3 eucalypt species. So that's Eucalyptus molucana, Eucalyptus fibrosa, also known as ironbark, and Eucalyptus terraticornis. The mid-story is dominated by Melaleuca decora, so that is a tea tree that can become dominant in frequently waterlogged sections of the woodland. And that is going to become important later down the track when I show you some data from that section. In the understory, we typically found Berseria spinosa, which is fun when you're doing field work in there, but also lots of ferns and grasses. And one final thing worth noting here is that there is a lot of mistletoe in the Cumberland Plain woodlands, and especially at our study site, and that goes in kind of boom and bust cycles. And mistletoe is actually an interesting case because they are hemiparasitic plants that rely on their host for structural support, and they rely on birds for distribution of seeds. So they have been known as a keystone species because they can boost biodiversity and they can accelerate nutrient cycling really well. They also provide food and habitat for many insects and birds that are depending on mistletoes for their survival. However, they can also compromise host health, and that is because they photosynthesize, but they obtain water and nutrients from their host. So they actually tap into the xylem and they just divert these resources towards them. And it happens that they have a generally higher water use and a fast leaf turnover. So they actually cycle through the nutrients from their hosts quite quickly. And a complicating factor is that if you are very fragmented woodland, then the birds don't have many opportunities to spread out. So some woodlands can get really heavily overburdened with mistletoes when they're very fragmented or when they're already degraded and the host defenses are down. So the problem with that is that parasitism actually can exaggerate water stress and carbon starvation for the trees when there is very severe infection rates at a tree. And if that's the case, then they can accelerate canopy dieback and they can even cause premature tree death. But it is worth to notice here that if the host dies, so does the mistletoe. So they definitely do not have an interest in killing their host. Okay, so the good and the bad sides of the mistletoe, they were actually really well known already. What I was interested in then was to find out how does this play out during drought. So how would the hosts cope with a leak in the water pipe during a drought, and are the mistletoes able to push them and to stress them as much so that they would be pushed to the edge with their high water use? So we have been monitoring mistletoe infection and host dynamics at our Cumberland Plain woodland via annual inventories at our TERN core hectare sites since 2015. So that is a 1 hectare plot where we monitor every tree for every year. And we also assess the mistletoe infection rates for every tree each year. So during that period, the mistletoe distribution had actually increased steadily until it peaked in 2018. So in 2018, about 70% of eucalypts in this 1 hectare plot were infected with mistletoe. So that's quite a severe infection rate, actually. And many trees were also quite severely overburdened, as you can see in the figure here. So that means that the mistletoes, they made up more than 60% of the tree's foliage, which is quite a lot, and you can see that it affected the trees across all size classes, but it especially affected the taller and the smaller trees at our study site. So with the onset of the drought, the mistletoe population started to decline, and then it almost entirely collapsed under the extreme conditions during the summer of 2019 and 2020. The recent drought, it was characterized by very hot and dry conditions. You can surely remember this was the case across most of the Australian continent, and our study site was no exception. So root zone soil moisture was frequently below average over the last 2 decades, and summers were getting increasingly hotter and the air was getting increasingly drier. And we had multiple heat waves that affected our study site during the tinderbox drought before rainfall events actually alleviated the soil moisture deficit at the early 2020. So as drought is typically accompanied by other stressors such as heat waves or diseases when the water stress trees are less resilient to biotic agents, I was very intrigued by the opportunity to study how the interplay of drought, of heat, and of biotic agent would affect carbon, water, and energy cycling. So I actually hired an arborist that you can see here in the left-hand picture. He's sitting there up in the canopy with an umbrella. And he climbed the tall trees. They were up to 25-meter tall. And he installed so-called set flow sensors in there. So I've shown you close up here. They are sensors to monitor tree water use continuously. And we had chosen branches that did have about 50% of mistletoe foliage attached to the branch, and branches that had no mistletoe. So we definitely wanted to make sure that we see an effect. And we then monitor transpiration dynamics continuously across these moderately infected trees over 2 summer seasons, and at Eucalyptus fibrosas, which is on the right-hand side, so that's the Eisenbach, and Eucalyptus molucana, so that we had a bit of a comparison how if there might be species differences that we can detect here. And yeah, we monitored this over 2 summers during the drought, and we were lucky enough to get a few heat waves in our monitoring period as well. So first off, I want to establish some baseline water use patterns that we can identify as like typical summer days that were neither too hot or too dry. And what I'm showing you here is diurnal water use patterns as they progress through the day on the x-axis, and how branch water use indicated here as transpiration changes throughout the day on the y-axis. So it's worth to notice here that transpiration rates were normalized by sapwood and leaf area so that they actually allow comparing the different branches, which obviously all had slightly different mistletoe to host foliage ratios. Alrighty, so what we see here is that these 2 species, they already had slightly different water use patterns even on normal days. So Eucalyptus molucana, that's the black line, they use slightly more water than Eucalyptus fibrosa in general. So that means fibrosas tended to be a bit more conservative in their water use. However, I wanted to know how mistletoe does modify branch water use here. And for both species, what we learned were that infected branches, they are indicated here by the dotted line and also colored by species. They had significantly larger transpiration rates compared to host branches. And that was even on typical summer days that were neither hot nor dry. Okay, so it's interesting to note here that the water use in infected branches from Eucalyptus fibrosa was far exceeding the water use of the infected branches and Eucalyptus molucana. So the water that the fibrosa was trying to save has actually been leaking out through the mistletoe in this case. So on summer days that were particularly dry, the uninfected trees, but also the infected branches, they both had slightly reduced transpiration rates when compared to baseline summer days, okay? This reduction was actually noticeable throughout the day, but particularly during the warmest hours in the afternoon. However, what we were really surprised to see was that during high temperatures that were associated with high atmospheric demand for water, then transpiration rates increased really starkly compared to typical or to dry summer days. And that was also the case irrespective if the hot days were also confounded by low soil moisture. So these notable differences the differences between the species were quite noticeable, but it was quite unexpected that the daily water use in infected branches increased about 2-fold in Eucalyptus molucana and about 4-fold in Eucalyptus fibrosa. So yeah, to recap, the mistletoes, they did indeed use more water in all conditions, but it was very unknown until then that their water use was especially large during hot days and during heat waves. And this study thus highlights that parasitic plants, they actually can substantially increase plant water stress when droughts are further compounded by heat stress. So the excessive water use of mistletoes was then adversely affecting the integrity of the host's water transport system. And we measured that as a loss in conductivity, and we actually found out that about 33% of the vessels were damaged in infected trees compared to 20% of, or less than 20% of vessel damage in uninfected trees. So here we could actually demonstrate that the compounding stresses of drought, of heat, and of mistletoe infection together, they did place the infected trees at greater risk of hydraulic failure. which is potentially further exaggerated if the drought would have gone on for longer, or if the mistletoe infection rates would be more severe. So it's then also wondering how would mistletoe presence and the hydraulic damage that they cause affect carbon sequestration rates at the standstill. And well, to find that out, we collected annual measurements of stem circumference in our 1 hectare plot, and we quantified the mistletoe load in the canopy in 20% increments. So that's what you're seeing here. 0 means no mistletoe, 5 means 80% of the foliage is mistletoe. And what we found was that stem growth tended to decrease with increasing mistletoe load in all sizes, but that there really was a threshold of about 60% mistletoe loading in the canopy when trees would become severely infected and they would stop growing. We then wanted to know if our plant and tree-based observations would match the ecosystem scale dynamics and how transferable our findings would be across sclerophyll woodland communities. So to answer that, we set up a paired flux tower experiment where we monitored ecosystem scale carbon, water, and energy exchange simultaneously in the 2 woodland stands during the drought. And one flux tower was located in our mistletoe infected section of the woodland, and another flux tower was positioned in a section of the forest that did not contain any mistletoes, and that was dominated mostly by melaleucas, who were otherwise forming part of the mid-story in the mistletoe infected eucalypt woodland portion. So what we're looking at here is latent heat flux, and that is the flux of heat from the Earth's surface to the atmosphere that is associated with evaporation or condensation of water vapor at the surface. So it kind of is an indication of evapotranspiration, and in our study here clearly confirmed that evapotranspiration increased when the air gets hotter. So that's indicated in the left-hand graph by increasing vapor pressure deficit on the x-axis, and that it decreases substantially when the soil dries out. So that's indicated on the right-hand graph with declining soil moisture on the x-axis. We also noticed that the latent heat flux was more sensitive to decreasing soil moisture than to hotter or drier air. And we also found out that the melaleuca stem seemed to lose more water than our eucalypt and mistletoe infected woodland even when it was hot or when the soil dried out. So that either indicates that they had access to more water in the soil, or that they were less restrictive in their water use and they risked greater hydraulic damage to sustain the high rates of carbon sequestration. And the picture that you've seen earlier here has actually been taken in this melaleuca woodland during the study. So you can see quite clearly how they were affected by the substantial canopy browning and canopy dieback during the drought. And that this actually suggests that they were not necessarily restricting their water use sufficiently to prevail through dry stress, and that they might struggle a lot more with more severe or more prolonged droughts. So yeah. To recap here, the drought is often accompanied by heat waves, and we don't really have a good understanding how both in isolation or in combination affect ecosystem resilience in the short or the long term. So the research that I presented, it really highlights that biotic agents can further compromise tree health and carbon and water cycling in already drought and heat stressed ecosystems, and that these agents might act as the final push to accelerate tree mortality. So while the explosion of mistletoe population, it had the potential to adversely compromise stand health at the forest that we had been measuring here, it's worth to notice that the vast majority of trees, they actually survived the drought. So they survived the heat waves and they survived the severe mistletoe loadings. And almost all trees had recovered very rapidly once the mistletoe population succumbed to the latest year of heat and drought stress, and once the favorable downpours at the end of the summer alleviated the drought. So I'd also like to point out here that the current focus on the mistletoe and host interaction has a bit distracted from the fact that the mistletoe population has also collapsed during heat and drought stress. So that indicates that they are very vulnerable, and that clearly has cascading effects down the interaction network and down for the species that depend on them for survival. So outside of our woodland, the mistletoe distributions, they were really reported to be increasing lately throughout southeastern Australia. And you all know that so is the likelihood of drought and heat stress. So we know that drought and heat stress can already weaken tree health, and that it makes hosts more, or trees in general, more susceptible to biotic disturbances. And our research here in Australia really falls in line with research from around the world, where mistletoes and biotic disturbances in general are emerging as important co-contributors to mortality in a warming world. And I want to conclude here by highlighting that the study hopefully clearly shows that when we're attempting to understand ecological responses to drought, that it is really important to monitor the effects of drought across scales. So I hope that I made a good case here for the importance of collecting ground-based validation data, and that we need to draw conclusions from multiple lines of evidence. So yeah, with that, I'd like to thank my collaborators, particularly Matthias, Elise, and Dan from Western Sydney University. And I'd also like to point out that the flux data is available at the AusFlux portal, and the inventory data is available at the TERN data portal for those that are interested. Thank you. And with that, I pass it on to Elisa.
Perfect, thanks. And I will be sharing my screen. And assuming there are no complaints, you can all see it. So we'll be talking about harmonizing ecological drought data on a global scale. And thanks to both Georgie and Anne for introducing a lot of information on drought. So this is going to make my job so much easier. So just to set the scene, I am going to start with the definition of ecological drought that was suggested by Crosby and colleagues. So they define ecological drought as a deficit in water availability that is episodic, and it drives ecosystems beyond their threshold of vulnerability. And this can then have impacts on ecosystem services, some of which may be relevant to humans. And these impacts can trigger feedback mechanisms, both in natural and human systems. So this graph here is very good at showing how the different components of this phenomenon interact with each other. So on the right-hand side, we have the natural side of the system in yellow, and on the left-hand side is the natural side of the system in yellow. On the right-hand side is the human side of the system in blue. And the idea is that there is a continuum between the natural components and the human components of this picture. At the top, we have the exposures. On the left-hand side, we have meteorological drought and the characteristics of the landscape. And on the right-hand side, we have anthropogenic-induced climate change and also the use of land and water resources by humans. So the next chunk of this picture is the sensitivity and the adaptive capacity of the different ecosystems. And this depends on the ecological characteristics of the ecosystem and also on the natural resource management strategies that are in place in that particular ecosystem. So when the balance between exposure, sensitivity, and adaptive capacity changes, then the ecosystem can be pushed over the threshold, and then we have an ecological drought. And this can create some impacts both on the natural component and on the human component. As I mentioned before, this is particularly the case when ecosystem services that are relevant for humans are affected. So these impacts can then trigger responses, feedback responses, both on the ecological side of things and on the human side of things. So the reason why this graph is particularly good is because it shows that ecological drought is a complex process that has different components interacting with each other. So we're talking about a complex process. There's many moving components, and there's also feedback mechanisms that further complicate the picture. And also another important thing to consider is that the characteristics of the ecosystems are really a key component in determining that balance between exposure, sensitivity, and adaptive capacity. And these further complicate the situation as well. So from a data analysis point of view, we know that the more complex your problem is, the more data you need. And in particular, in this case, we really want as many data as possible. ideally distributed across as many ecosystems as we possibly can all around the globe, and we like to have long-term data sets. So where are currently these data? Well, typically they are saved in national data repositories. So these are managed by ecosystem research infrastructures such as TERN. These are national networks that collect ecological data in a standardized way, and then they make this data available on their data repositories. So this map shows the different sites where TERN is collecting data across Australia. It's over 800 sites. The different symbols on the map correspond to different types of data that are collected at each site. And these data are then all collated and made available on the TERN data repository, where the user can search and query the data, and they can download the data and then use them for research or environmental management purposes. So TERN is not the only ecosystem research infrastructure. There's other similar infrastructures around the globe. And a few years ago, 6 of these infrastructures have come together to create the Global Ecosystem Research Infrastructure, also known as GERI. So at the moment, GERI consists of TERN, which represents Australia with over 800 sites, the National Ecological Observatory Network, also known as NEON, from the US, with over 80 sites. There's then a couple of infrastructure from Europe, which is 650 sites. And then there is the Chinese and the South African networks, which have about 40 sites each. So all in all, GERI has over 1,600 sites across the globe. So, oops, sorry, too fast. Recently, GERI has been awarded a NASALNet grant to combine the ecological drought-related data on a global scale. So the data from across all the different networks are going to be combined into one data repository. So this is a NASALNet grant, which is a program funded by the National Science Foundation based in the US. And its aim is to increase the collaborations between the different networks. And the purpose is to speed up the progress in a particular area of science. And since drought has been identified as an environmental priority, GERI had the opportunity to work on this project where we're trying to bring together the drought-related data from across the globe. So I mentioned before that within each national network, the data are collected in a standardized way. However, the standards may be different between the different networks. And this can be for a variety of reasons. For example, the type of methodology or the type of sensors that are used to collect a particular type of data may be particularly suitable for the predominant ecosystem types of that particular country. And another country may be better off using different methodology or sensors, or it can be for historical reasons. So, for example, if the majority of the scientists within a certain country have agreed on a standardized methodology, then the country is then going ahead and follow that particular one, but the researchers in a different country might have gone a different way. So that's why the data are collected following different standards between different networks. So we cannot standardize the data because they have already been collected. However, the project is now aiming at harmonizing the data. So data that have been collected with different standards, how can we merge them together so that they are research ready? Now we are in the first phase of this project. It's currently ongoing, and that consists of harmonizing the data between TERN, which is the Australian partner, and NEO, which is the US partner. And the second phase, which is going to kick off in a few months, will be extending the data harmonization to the other national networks within GERI. And this is going to be a little bit of a proof of concept or a guinea pig, if you want, to then tackle other data sets of ecological importance. So what are these data that we're looking at? So at the moment, we're working on 6 base data products. We are looking at precipitation, air temperature, the water content, temperature and texture of the soil, and also we're looking at a couple of measurements of soil biomass, in particular soil microbe and soil roots. So the first 4 of these measurements, they come from flux towers. So Anne mentioned the flux towers in her presentation as well. These are infrastructures where the sensors that collect different types of environmental data are mounted on. Typically, the top of the tower sits on top of the canopy cover. So the flux tower has different heights depending on the type of vegetation that is characteristics of that environment. So the characteristics of the flux towers is that they collect continuous data. The frequency of data collection can be every second, every 10 seconds, every minute, depending, or anything along those lines, depending on the setup. However, the idea is that we have long-term continuous data coming in. The other types of data, the soil texture and the soil biomass, they tend to be one-off measurements. They're typically done at first when the flux tower is installed or soon afterwards, and only repeated occasionally every few years. And that's because this particular type of data, they don't change quickly with time, so we don't need to repeat the measurements as frequently. So how are these data from the different national networks going to come together? So these data, as I mentioned before, are saved on the individual national repositories. So they're represented here in green by these green cylinders. Each one of them is a data repositories of one of the 6 national networks. And the systems that we are working on at the moment is going to fetch the data from the individual data repositories as well as the related metadata records, and it's going to import them into a central repository, essentially a database. And within this repository, then the data is going to be harmonized. So the data coming in with different standards or formats is going to be all harmonized together, so it can be merged and it can be analysis ready. The metadata record is going to have to undergo a similar revision so that it's all compatible. This may involve some transformation of the data in the metadata record, and we're also considering creating the right products, such as calculation of risk of drought at the different sites across the network. So all of this data will then be made available through a portal where the users can search and query the data, and they can download and use the data. So while we're talking about data harmonizations, what are we dealing with? So these are the challenges that we are working on right now. It's just a few examples of the current work that we're doing, and I'm expecting that as we include more of the networks into the global repository, we will have more challenges coming our way. So at the moment, we are dealing with differences in methodology or in the type of sensors that have been used to collect the data, the location of the sensors, especially the sensors are mounted on the tower or the soil probes. They might have been mounted at different heights, or they may be going down at different depths. The frequency of the sampling is different between networks, and also the procedures that we use for quality control and gap filling of the data. The units of measurements are different between the networks and also sometimes within networks, and this may come as a surprise. This is typically the case for the soil texture data, that they may be expressed as percentages or as grams or milligrams per unit of volume. So that will need to be all standardized. We have some missing data sets, so some of the data are not available at all the sites. So we are considering whether we have other data sets available that we can use to fill in the gaps. And we also have difference in formats and also the content of the metadata records that we'll need to standardize. So as I mentioned before, these are just a few examples, but I'm sure that the list will grow longer as we keep working on this. So just to summarize, this is the first example of collating drought-related data on a global scale. And as far as I know, one of the few examples that are merging together environmental data across the planet. The main thing that we're learning is how to do this data harmonization to put together data collected by different networks. And we want to use this knowledge to then, going into the future, being able to use a similar process to bring together other environmental data sets that have been identified as priorities. So this is me, and I will stop sharing, and then I will hand back over to Beryl.
Oh, that was fabulous. Thank you so much, Elisa and Anne and Georgie. I think we've all been privileged to hear such interesting facets of the question of drought. I'll get everyone to turn their cameras on now on the panel. So Elisa and Anne, if you can come back, so that we can answer some of the questions. So Elisa, since you were the most recent speaker, you talked a lot about standardizing the data across all the networks in the world and the aim of harmonizing. What's our success metric for how will we know it's been harmonized and whether that's been beneficial? Have you got any ideas?
That's a very good question. And it's always the case when putting together this type of infrastructures where we are collecting and collating the data to make them available for someone else to use. We really need this system to be tested by the researchers that are going to use the data. So at the moment, the plan is for researchers involved in ecological drought research, and in particular, the researchers from the Sullivan Lab at Oregon State University to test the beta version of the infrastructure. And then based on their feedback, we will adjust what needs to be adjusted and make the necessary improvements to go on into the future.
Thanks, Elisa. I've got a question for Anne, but there's one in the Q&A that I think I'll take from Alexandra, who's asking about the group that the observatories are actually covered in the global ecosystem research infrastructure and why there isn't Central or South America. It is a plan, Alexandra, to do that. And in fact, there's a big meeting on the 5th of December in Brisbane to talk about how we can widen the participation. And Elisa, I think you've been helping the Jerry partners work with Mexico to try to gather more information from Mexico, and there was a plan to bring South America into it. So the global ecosystem research infrastructure at the moment represents the more continental scale observatories, but there's going to be a subsection of it called GENI, which is a global ecosystem network of networks infrastructure, which will allow us to bring in more country groups. So great question, Alexandra. And Anne, the question that I got for you was somebody's read a paper that says that when people do vegetation restoration projects, they don't do the parasites, they don't bring in the mistletoes. And you indicated in your talk that mistletoe is both heat stressed and also vulnerable to drought. And so if we've got major restoration projects that miss those things, and apparently the restoration projects have shown that you do need the parasites as part of the whole ecosystem and because of everything that feeds on them. How's that going? Do you think you need to now set up a site in a restoration area to test some of your data, is the question, if it's not too hard?
Yeah, no worries. That's a good question, actually. I think what I do just want to clarify here that I did give mistletoes a bit of a bad rap because we were focusing on the physiological interactions with their host at a very, very intensely impacted site. So usually the sites are not as impacted, and that's when they really become important for restoration projects for all the reasons that Beryl mentioned and for the ones that I've highlighted before, that they are really boosting biodiversity and they are a keystone species. So yes, I'd say it's definitely important to include them in a restoration project and to account for them when doing assessments of restoration sites. Regarding monitoring, it would be very interesting to monitor the dynamics if they're also as vulnerable, if there aren't as many mistletoes on a tree. So what we had observed at our site was that almost all mistletoes died. So there were very, very few, like very isolated. As I said before, 70% were infected before. And we really had to look around and find isolated mistletoe plants that did survive the heat and the drought. So it would be actually quite interesting to see if the site hasn't been as infected and the trees hadn't been as stressed, if that would result in higher survival rates for mistletoes and thus preserving these ecosystem services that they provide. So yeah, it would definitely be interesting to do a follow-up study for a restoration project.
Thank you, Anne. And we have actually reached the end of our time allotment, and there's still some questions in the Q&A, but for those that have put them there, be assured we'll be back in touch with you to follow that up. And I think I just skipped the valuable slide that I wanted to show you, and that is that we're back next year. We have bi-monthly Wednesday webinars, and we expect to see you all there. Thank you. The first one is actually that difficult question in March about how much do you confound your ecosystem monitoring if you put site experiments there, because there are a whole lot of people that say that we're at the point of urgency where we actually need to do a little bit of experimentation alongside our observations to hurry us up. So please come along and help us participate in that. Thank you very much, speakers. I think that the comments in the chat show how much everyone has enjoyed it. Thank you very much, everyone who came along to listen and ask questions and just watch today. And we look forward to seeing you in 2025. Thank you.